Isha, go ahead.
Yes. Okay. So welcome everyone. It's my honor to introduce Dr. Raj Kothapalli, who's an associate professor at the Pennsylvania State University's Department of Biomedical Engineering and Penn State Cancer Institute. So just give me a moment. Please let me know if you can see the screen. So this is the IASNM Radiant Flashpoint Educational Web Series in collaboration with Missy. So today we have here Dr. Raj Kothapalli, as I said, who is an associate professor at the Pennsylvania State University's Department of Biomedical Engineering and Penn State Cancer Institute. His journey began with a PhD in biomedical engineering from Washington University in St. Louis, where he was trained under the guidance of Dr. Liu Wang. Following this, he has pursued a postdoctoral training at Stanford University, their molecular imaging program under mentorship of Dr. Sam Gambhir. So Dr. Kothapalli's dedication to this academia led him to serve as an instructor in the Department of Radiology at Stanford University. So he's for assuming the leadership of the Biophotonics and Ultrasound Imaging Lab at Penn State in 2017. His work focuses on developing innovative multimodal imaging strategies, blending optical and ultrasound and photoacoustic technologies. Dr. Raj's contributions have been recognized with the prestigious awards, including the Hamiltonian Peter Michael Postdoctoral Fellowship, the K99R00 NIH Pathway to Independence Award, and the NSF Carrot Award. I'm sorry there. So by congratulating him on his achievements, please join me in welcoming Dr. Raj Kothapalli for his talk, which is titled as Lights, Sound, Action, Multimodal Ultrasound and Photoacoustic Imaging Strategies for Preclinical and Clinical Applications. So before we go into this, if you have any questions while the session, please put it in the chat box, or you can kindly raise your hand and post the speech. You can unmute yourself and yeah, we'll answer your questions. So over to you, Dr. Raj.
Thank you, Isha, for such a nice introduction, and good evening, and for folks from India, and good morning if you are joining us from East Coast or West Coast. Yeah, let me. Oh, God. Okay, so the title of my talk is 1 second. Why this is or maybe I can move this to another place. Let's see a cursor. Okay, so today I'm going to discuss about my lab's research in putting together multimodal imaging systems that combines optical, ultrasound, and photoacoustic imaging technologies and their applications for preclinical and clinical applications in covering wide range of diseases like cancer, cardiovascular, as well as neurological diseases. So we are going to also, I'm going to also discuss what is the motivation for combining these technologies. And this is my contents of today's talk: an introduction to photoacoustic imaging and challenges, and some applications of multimodal ultrasound and photoacoustic imaging, in particular prostate cancer, brain cancer, and peripheral arterial disease. This is a project that we are working with Indian Institute of Science, Bangalore, the PAD, and then some future direction. These future directions are mostly geared towards the technological innovations, not necessarily on the application based. So if you look at the whole body multimodal imaging, so this slides actually doesn't need introduction to you because you are most of them are medical background. So if you look at the multimodal whole body imaging, so we have a PET imaging, which is very highly sensitive molecular imaging technology. You have to induce radioactive tracers that target different types of biomarkers. So the most popular being the FDG PET targeting the glucose metabolism and for imaging the tumor activity. So that's what you see here. This is the whole body PET imaging. And then you have a X-ray computed tomography that provides the anatomical information. And in oncology, this PET-CT is widely used to provide both anatomical and molecular information together. If you just display the molecular information, you would not know what is the origin of that molecular information, which organ you are getting that molecular information. So by combining this anatomical and molecular information, so you have that more accurate localization of the molecular information. So, but one of the main limitations of this PET-CT is high cost and maintenance, and also not suitable for point of care applications. I'm wise enough to say not other limitations such as ionizing radiation for this audience because you work with this radiation. But one of the limiting factors is also the use of ionizing radiation in both the cases. So on the other hand, we have point of care ultrasound. For example, we have ultrasound in medical emergencies like in ambulances, right, for assessing the acute heart failure. So they use a point of care ultrasound that can be connected to a cell phone where you can image the Doppler ultrasound as well as regular B mode ultrasound to evaluate the vascular functions such as this includes actually not just for heart failure, but also for lung ultrasound. You can assess the accumulation of the fluids using these ultrasound devices, point of care ultrasound devices. If you look at the other applications of this ultrasound, they have this emerging technology called functional ultrasound that you could use it to image the brain activity of newborn babies. They have skull is not yet formed in this newborn babies. They have this transfrontal regions where you can shine ultrasound through, and you can acquire some high resolution ultrasound images. This is conventional B mode ultrasound image, which provides you the structure of the brain. But you also can enable power Doppler ultrasound, which is actually uses ultra-fast ultrasound based imaging strategy to map microvascular blood flow, which is slow moving blood flow inside the brain regions. So for the last several years, this technology, this power Doppler ultrasound, people also call it functional ultrasound imaging, is gaining a lot of importance for point of care applications such as in neonatal ultrasound units. So, but the problem with ultrasound is that it is sensitive to blood flow. It can provide functional blood flow information, but sometimes this is operator dependent, and that's one limitation. And other major limitation is it's not molecular in nature. So it doesn't provide you a direct molecular information. So most of the diseases, when they are at early stage, the changes that are happening are at molecular level. So we need more sensitive technologies like PET imaging, which is very sensitive to molecular imaging. We need, but PET has limitations of high cost, high maintenance. But we need some technologies that are very sensitive to the molecular information. So that leads to the motivation for using the optical radiation. If you look at this electromagnetic spectrums, like, we have gamma rays that are used in PET imaging, X-rays that are used in X-ray CT. But if you further go towards the shorter, higher wavelengths, right? So then you have this visible spectrum or near infrared optical spectrum. So here, unlike X-rays, which uses hundreds of kilo electron volts, so the optical radiation, the optical photon has a typical energy of 2 electron volts. And the light interaction with the tissue can be at molecular level and it can lead to functional information. For example, oxygen saturation, because oxy and deoxyhemoglobin absorb light differently, uh, these two different molecules, and it can lead to functional oxygen saturation information, or it can lead to metabolic information. Like, for example, if you're imaging certain autofluorescent molecules such as NADH or ADH, they can provide you a direct fluorescence-based metabolic information. Or it can also lead to biomarkers, cell-specific biomarkers. If you are taking fluorescent molecules and you're tagging them with certain peptides and you are able to target certain biomarkers highly expressed in the cancer cells, it can also provide a direct cell-specific biomarker information. Optical imaging is scalable. You can have a microscopic techniques, you can have endoscopic techniques, you can also even develop macroscopic tomography-based imaging techniques. So as you can see, so your spatial resolution varies, your spatial resolution as well as imaging depth changes from as you scale the technology from microscopic to macroscopic regimes. So there are wide variety of optical imaging systems. The bright field or wide field optical imaging system, fluorescence imaging. There are wide variety of fluorescence imaging systems. Even you have optical coherence tomography systems and emerging photoacoustic technologies that provide a rich. The one take home message here is even without injecting any contrast agent, you can get a rich, high contrast information from the tissue, which is completely different from a B-mode ultrasound or other imaging technologies such as X-ray, CT, or PET image. So, but there are fundamental challenges with optical imaging. One is molecular specificity. There is this paper during COVID, everybody is using these pulse oximeters to monitor the oxygen saturation. There's this paper in New England, published in New England Journal of Medicine, talking about racial bias in pulse oximeters, because light has to travel through the skin, and the skin color actually affects the quantitative accuracy of oxygen saturation. Right? Because you have so many types of molecules within the tissue. So the one skin layer is dominantly present with this melanin molecules, and then you have oxy and deoxyhemoglobins and other molecules. So our solution to this is, right, you need to incorporate some model-based simulations to understand how light interacts with different molecules. And these model-based simulations and photon propagation models, right, has to be incorporated with this kind of devices to accurately quantify the oxygen saturation. So there is this molecular specificity issue with the optical imaging, especially as you want to image deeper inside the tissue, this problem becomes more challenging. So other challenge with optical imaging is the resolution. Light, unlike X-ray photons, light doesn't travel like a straight path. Light gets scattered a lot inside the tissue. For example, if you are only imaging superficial tissue, such as 500 micron, it's the diffraction that limits the spatial resolution, which was overcome using super resolution methods, which can provide 20 nanometer resolution as opposed to 200 nanometer resolutions, which conventional optical microscopy techniques can provide. But if you want to image deeper, further deeper, like beyond 1 millimeter to up to several centimeters into the tissue, then the light diffusion limits the resolution capabilities. This slide actually kind of, this little experiment summarizes what is light diffusion. Let's say if you are shining the light through a clean water, right? So you can see this is a light from a laser pen. So you can see that laser spot on the back wall. But if I add few drops of milk into the water, the light start to scatter a lot, right? So if I add more milk, then light scatters more inside the tissue. You don't see any photons escaping here with your naked eye. But if you switch off the lights and if you put highly sensitive light detectors, you can even see those photons, but you would not know where those photons are coming from inside the medium. right? So people in conventional optical imaging, they put bunch of light detectors or fibers, and they use rigorous mathematical reconstruction algorithms, but still they would end up, having a poor resolution, typically about a 1/5 of an imaging depth. Let's say you're imaging 5 centimeters deep a region, so then the typical resolution is 1 centimeter. So then the question is it possible to generate high contrast and high resolution images of deep tissue, although the light gets scattered a lot like what you see here? So for that, we use photoacoustic imaging technologies. Photoacoustic is a hybrid imaging technology where you shine the light into the tissue. Light gets absorbed by the molecules such as hemoglobin or melanin or water or certain lipids. So these molecules undergo thermoelastic expansion. They generate, a transient heat of order of millikelvin, and this will, in fact, manifest into ultrasound pressure waves, and they propagate outside the body, and they can be detected by conventional ultrasound transducers. So the light absorption of molecules resulting into ultrasound waves, right? That's what is called photoacoustic effect. And it's nothing new. Actually, Graham Bell discovered this photoacoustic effect at 1880. Although he doesn't have lasers, he was using sunlight to demonstrate this effect. And if you're still confused, what is photoacoustic effect? The thunder that you hear from lightning is actually a photoacoustic effect. But here, those gigantic electromagnetic waves are striking the aerosols, which are big particles, and creating this audible sound. But in photoacoustic effect that we use in biomedical applications, our lasers are very, generate tiny pulses, and the molecules are also very tiny. So therefore, I say that they don't generate a audible sound, but an ultrasound, tiny ultrasound waves, right? So in compared to optical imaging, because in photoacoustic imaging, you are getting the contrast from the molecule, because only those molecules that absorb light generate the photoacoustics. By tuning the wavelength of light, and which molecule absorbs that light, and you can precisely tell using spectral unmixing or other techniques what molecules are generating that signal, right? Therefore, you get the contrast from the light absorption, but the resolution and the imaging depth is actually also controlled by the ultrasound. Unlike light, ultrasound is not scattered as much. As you know, that ultrasound can be used for imaging even babies inside the pregnant women, right? So ultrasound doesn't have a problem of traveling deep inside the tissue, or able to localize the ultrasound is not a problem. Therefore, you are gaining the advantage, because once the light converts to a sound, and that sound waves doesn't get attenuated as much as light waves attenuate inside a tissue. Therefore, you have a cleaner detection that improves the spatial resolution and imaging depth. Therefore, compared to the conventional optical imaging, so we have a better spatial resolution and imaging depth with photoacoustics. And there are different types of molecules that absorb light, and these are intrinsic chromophores like melanin, which is responsible for melanoma cancer. You can detect that melanoma cancer label-free using photoacoustics. There are many clinical studies that are ongoing right now with this technology. And oxy and deoxyhemoglobin, because they absorb, see, as you can see here, so the red one is a deoxyhemoglobin absorption as a function of wavelength, and blue curve is a absorption of an oxyhemoglobin as a function of wavelength. So by shining multiple wavelengths on the tissue, I can quantify the oxygen saturation. How much is oxyhemoglobin present at a particular vessel, or how much of a deoxyhemoglobin present, and based on the ratiometric analysis, I can quantify the oxygen saturation. That is the fundamental strengths of photoacoustic imaging, label-free imaging of oxygen saturation that no other technology can do. There's a fMRI that can provide, bold MRI can provide oxygen saturation, but it's an indirect information, and it's not at high resolution as what photoacoustic can provide. So that's the fundamental strength of photoacoustic imaging, of label-free imaging of vascular structure, as well as oxygen saturation. That itself has many applications, not just for cancer, because cancers are supposed to be hypoxic, And and you can detect the oxygen saturation of the tumors. And you can also use this information to study brain activity. So you have higher brain activity, then you have more hemodynamic, activity in those regions. So you can use this technology to study brain activity and also for vascular diseases in general. So not just this intrinsic chromophores, like in PET imaging or MRI, you can develop a molecular imaging agents. That means you can use extrinsic contrast agents. Anything that absorbs light, you can use them, or you can adapt them for photoacoustic imaging. For example, a wide variety of fluorophores, right? So you have those fluorescence agents. In fact, a bad fluorophore is a good photoacoustic agent. If you look at the molecular excitation, right? So once a molecule absorbs the photon, it has two options when to decay from excited state to ground state. One option is to by emitting fluorescence. Other option is to emit heat. Both are competing mechanisms. So if the molecule is emitting less fluorescence, that means rest of the energy is transferred to the heat, right? So that is good for photoacoustic. The more heat the molecule can generate, so there's a more photoacoustic generation. So therefore, you can use the existing fluorophores or even more specific molecules or nanoparticles that absorb light. For example, gold nanoparticles, carbon nanotubes have been widely explored for photoacoustic as photoacoustic contrast agents. And you can do multispectral imaging to further understand the distribution of this contrast agent. So it's a photoacoustic imaging is a multi-scale technology. You can have a photoacoustic, the way you arrange the light and sound, right? And depending on what is your targeted imaging depth and spatial resolution, right? So once you fix those, you can actually develop a photoacoustic system for that application. So it could be a photoacoustic microscope where light and sound are focused tightly to a point, and you can get a sub-micron resolution. As you can see here, this is an image of a DNA and RNA of inside the cells. Because DNA and RNA can absorb UV light and they generate photoacoustic sound, you can actually map the DNA and RNA without inducing any without staining the tissue. Normally in conventional pathology, so they stain the tissue to see internal cellular structures. But here, because these molecules absorb light, you can image them label free. This is an image of a melanin. And here, this is the image of a mouse vasculature where the red pixels are showing highly oxygenated blood, and the blue pixels are showing the low oxygenated blood. If you sacrifice the resolution, you can further image deep. This is the photoacoustic acoustic resolution photoacoustic, as they call it. Here you are able to penetrate up to a few millimeters deep. In the microscopy, this is called optical resolution photoacoustic microscopy, the top row. So here the penetration depth is less than a millimeter. But in the acoustic resolution photoacoustic microscope, you can penetrate up to 5 millimeters depth, but you have to sacrifice the resolution. The typical resolution here is about 30 to 50 microns, right? Here, this, for example, besides the vasculature imaging that is coming from hemoglobin absorption, you can also image gold nanoparticles. In this example, they injected a gold nanoparticles that targets the cancer region. So you can clearly delineate the contrast from the gold nanoparticles compared to the rest of the background. So if you further sacrifice the resolution, right, so you can image further deep. For example, you can now envision taking a conventional ultrasound probe and attaching light guides next to it. And now you can do photoacoustic imaging. In fact, combined ultrasound and photoacoustic imaging, because you have a traditional ultrasound probe to which you are coupling the light, right? So you can do both ultrasound and photoacoustic imaging together. So for example, here in this image, this is actually from a breast cancer patient. So where they injected a methylene blue dye that actually maps the sentinel lymph node in breast cancer patients. So they are showing that the contrast coming from the methylene blue dye. So it has a lot of applications. For example, for identifying sentinel lymph node is very challenging for in these breast cancer patients. Normally, they open the breast and after injecting the dye, so the surgeon opens the breast and looks for the contrast from the blue dye. But here, using minimally invasive surgeries, you can accurately pinpoint where the dye is, so that you don't have to do that kind of open surgeries. So it becomes a minimally invasive surgery if you use a photoacoustic imaging guidance. So you can also develop some endoscopes. As you can see here, you can develop even a 2 or 3 millimeter endoscope where light and sound are integrated within that little 2 or 3 millimeter device, and you can image the intravascular ultrasound. So you can actually map the vasculature inside a colon or other internal structures. So there are many emerging clinical applications in particular, right? So this is, for example, one application from my advisor, Li Hong Wang's lab at Caltech. So where they have a dedicated photoacoustic tomography system for imaging human breast. This is the label-free imaging of breast vasculature. And they have done like several studies to show that based on the hypoxia as well as photoacoustic elastography information, they're able to image the breast tumors. So in particular, my work in cancer is focused on detecting human prostate cancer. This dates back to my postdoctoral fellowship under the guidance of Dr. Sam Gambhir in Stanford molecular imaging program. So I started in 2009 developing this combined transrectal ultrasound and photoacoustic imaging system. So it took me 4 years to develop this device. So using a novel ultrasound transducer technologies, and then further 2-3 years to translate this into a urology clinic and study like 20 patients, including 10 patients for the first time who have been imaged with intravenous ICG. ICG is a fluorescent molecule which is clinically approved for several applications, including brain cancer detection, kidney partial nephrectomy. There are a lot of applications of fluorescence-based imaging with ICG molecule, but we don't have a dedicated contrast agent for photoacoustic imaging. So therefore, we used ICG, which in fact is a better photoacoustic agent because it has a less quantum yield of only 10% quantum yield. That means you can assume that rest of the energy is transferred to heat and can generate a very good photoacoustic signal. So, so therefore, we injected ICG into the patients while we are doing transrectal ultrasound and photoacoustic imaging. I'm going to show some results. But once you develop, the motivation actually comes from the fact that transrectal ultrasound is the gold standard for detecting the prostate cancer, but it doesn't detect the cancer, but it is used for guiding the needle biopsy. So that there are about 10 to 12 biopsies taken randomly. They are not targeted biopsies. They are randomly chosen within the prostate volume, and these biopsies are sent to pathology to see if there is a cancer or not, right? But because the nature of the biopsy is blind, which is not targeted, so there is a high chance of missing the prostate cancer. So our idea is to integrate the light fibers into the same transrectal ultrasound and now do multimodal ultrasound based structural information as well as photoacoustic based molecular information, so that that molecular information can pinpoint where exactly the cancer is, and you can use then that for a targeted biopsy. So with that motivation, I started working on developing that system, and this was the system I started with the benchtop system, and then this is the system which I integrated by by 2013 or so, and which was placed next to the patient bed in the urology clinic at Stanford, and we imaged like 20 patients, as I mentioned before. And as I said, this uses very novel ultrasound transducers like capacitive micromachine ultrasound transducers that are sourced from Dr. Kuryakub lab at Stanford, who is my other advisor in the electrical engineering department. So in collaboration with their lab, So we developed this device. And I'm not going to bore you with a lot of details, but whenever you develop such a new system, you have to convince yourself and to the physicians it can penetrate deeper. So we did some tissue mimicking phantom studies. We also extracted, not we as in like for our study, but these patients who undergo radical prostatectomy as a standard of care. So we got the prostates from these patients, the whole intact prostates, and we put some artificial blood tubes to convince ourselves that we are able to penetrate deeper using our imaging systems, right? So this is one such result. And we did a lot of ex vivo studies to see, we imaged about like 20 to 25 ex vivo prostates. So the more exciting results are here in the in vivo. So we eventually inserted the device into the patient rectum, and we imaged the human prostate using this multimodal ultrasound and photoacoustic device. As you can see, this left one, this is an image of a ultrasound image of the prostate, and the right one is the photoacoustic imaging of the prostate, and then the middle is the ultrasound co-registered photoacoustic image. Right, all of them are displayed in real time. Ultrasound frames are 30 frames per second. Photoacoustic frame rate is about 10 frames per second, mostly limited by the laser firing rate. So our lasers that I use for this deep tissue imaging can fire up to only 10 pulses in 1 second. So we can have a 10 frames per second. So this co-registered frame is also updated about 10 frames per second. So because we are doing label-free imaging, so in photoacoustic image, in the ultrasound image, you can identify several structures, right? The prostate, peripheral zone, transitional zone. This is the bladder, which is more hypoechoic because it's filled with water. And we can identify typical structures in the ultrasound. But photoacoustic, because it's only sensitive to the light absorption of the molecules, so we have a dominant contrast from wherever the light is being absorbed by the blood vessels. So one particular blood vessel that we used to see very often is the neurovascular bundle. And we also used to see several other major vascular structures such as DVC, dorsal venous complex, which is a major 2 millimeter or few millimeters kind of a diameter blood vessels that covers from peripheral zone to the transitional zone. So we see dominant contrast from the DVC as well. And some cases we have seen this kind of abnormal contrast, which we could be because of the tissue being stiffer there, or it could be indicating a presence of cancer, like what you see here, right? Which cannot be diagnosed just based on the abnormal photoacoustic contrast. That's where you need to inject certain dyes or molecular imaging agents to understand or to get more specific information about why you see such an abnormal contrast in certain regions, right? So that's what we did. For that, as I said, right, so we would like to use ICG and to see if it has a preferential uptake in the tumors. First, we wanted to, before we inject into humans, right, so we wanted to convince ourselves, can we do this in mouse models of cancer, right? So here is one example. There is a PC3 cancer cell line is injected into the mouse, forming a subcutaneous tumor. You can see the bioluminescence imaging confirming that there is a cancer there, right? And then the same device which we are supposed to use in humans, here I coupled that from the top surface to the tumor, and I can see that, before injecting the ICG, you see the tumor region only shows the contrast from the blood, but after I inject the ICG intravenously into the tail vein, that ICG is leaking into the tumor regions through the leaky vasculature. You see some enhanced contrast, right? So this is at a 800 nanometer wavelength where the ICG has a dominant absorption. But we want to use more analysis to confirm there is ICG. So we also did spectral analysis. As you can see from pre-ICG injection compared to the post-ICG injection, you see the difference in the spectral trends for the certain regions of interest. So that also confirms the ICG. We also further did spectral unmixing to confirm that the tumor regions have the ICG compared to this is the pre-injection tumor imaging. This is post-ICG injection tumor imaging, and you can see that ICG is leaked into the tumor region. So after validating our analysis and methods in mouse models, we did the same thing for the first time in the these are the first in human studies, as I mentioned before. So we did the same thing in the human patients. For this initial study, we targeted certain patients who have cancer in the peripheral zone that we can easily see. This is like multimodal imaging, where the MRI has been confirmed, the preoperative MRI, like our pre-procedural MRI, shows that this men have some abnormal, MRI contrast, and we know that this is the tumor region. And then we directed our ultrasound probe towards this region, and then we injected the ICG and to confirm that. So as you can see here, this is the pre-ICG from this region, and then this is the post-ICG unmixed image, and we could confirm that there is a ICG leakage into the tumor. And also the spectral analysis also confirms that there is a presence of ICG. So this is like very preliminary studies. Now we are funded by NIH R01 grant, just came through 6 months ago. This is in collaboration with the University of Michigan. So we are going to image 50 patients in Hershey College of Medicine and 50 patients in the University of Michigan to further study ICG-based prostate cancer detection using photoacoustic imaging. So because the ICG has a problem of staining the vasculature unspecifically, so we are aware of that problem. So therefore, we started working on dual modality ultrasound and photoacoustic contrast agents. This actually is coming from my K99 R00 grant. I started this work in Stanford in 2013-ish. So these are the results from that work. And right now we have a DoD grant to further improve these results. So what we did in this, the idea here is to encapsulate ICG inside the microbubbles. Microbubbles are traditional ultrasound contrast agents, and people are also even developing nanobubbles. For example, we have a collaboration with Agatha Exner's lab in Case Western University, where they develop these nanobubbles. So our idea is to encapsulate this ICG inside the microbubbles as well as nanobubbles, and use this dual modality contrast agents to further enhance the prostate cancer detection, because our device can generate both ultrasound and photoacoustic contrast. So then our rationale is that why not we use contrast agents that also can provide both ultrasound contrast as well as photoacoustic contrast. So although there are many groups developing different types of contrast agents, but we focused on microbubble based dual modality ICG and microbubble based contrast agents, because both microbubble and ICG are clinically approved for ultrasound as well as for optical imaging. So therefore, we thought it's easy to convince the FDA eventually to, if you are using this clinically approved contrast agents. So here, as you can see, this is a TM image of a nanobubbles loaded with ICG, and which confirms that these particles are 200 nanometer size. And we did a little phantom study to where we have made some wells, and this is an agarose phantom, and this agarose phantom is loaded with bubbles. For example, here the top row is nanobubbles loaded with ICG, and the second row is just the nanobubbles, there is no ICG, and the third row is microbubbles loaded with ICG, and the fourth row is just the microbubbles. As we can see that, so you don't see photoacoustic contrast from the nanobubbles, just the pure nanobubbles are pure microbubbles. But from the nanobubble loaded ICG and microbubble loaded ICG, you have more photoacoustic contrast agents. So why we tried both nanobubble loaded ICG as well as microbubble loaded ICG is because microbubbles have this, because they are a few microns in size, they don't leave the vasculature. So if you want to target tumor microenvironment or cell-specific biomarkers, then you need contrast agents that can extravasate out of the vasculature and can go and target the cells. Therefore, we also explore nanobubble-based ICG agents. And I did some preliminary studies back in 2014. Unfortunately, this study is not yet published. We are hoping to get more results. So here, um, uh, we formed the subcutaneous tumor, and in this case, on the left side, I injected nanobubble-based ICG into the tail vein, and these are not targeted agents. So they are just like, we are looking at how the EPR effect, enhanced permeability, whether it can help us see the tumor more clearly. As you can see, we did longitudinal imaging of this mice with photoacoustic imaging. So as you can see, at 60-minute time point, we have a higher uptake of this nanobubble-based ICG. So the microbubble-based ICG loaded with microbubbles inside the microbubbles did not provide us that much contrast compared to the ICG loaded inside the nanobubbles. And as I said, this is now funded by DoD grant and in collaboration with Dr. Agatha Exner and Dr. Gail Matters at Hershey College of Medicine. And Agatha is from Case Western University, Cleveland Case Western University. And we are actually further optimizing these contrast agents to specifically target prostate cancer cells using biomarkers such as PSMA or bombacin. And this is an ongoing study. So, and we are also funded by NIH to develop or to further improve the instrument capabilities. What we envision is to incorporate more imaging features into this technology. So far I was telling only B-mode ultrasound and photoacoustic based vasculature imaging or ICG imaging. But in fact, you can also enable here other capabilities such as Doppler ultrasound, or in fact, power Doppler ultrasound, which can even provide a finer vasculature, and also ultrasound elastography that can provide the elasticity information of the tumor, right? So therefore, we tested those using a multimodal contrast phantom. That what you see here is a complex phantom, which can provide all these different types of contrast. So therefore, we tested that in this phantom. As you can see, this is an ultrasound image. You can see the bladder region. You can see a prostate region. This is the prostate. This is the bladder. Bladder is a balloon filled with water, and this is a custom-made phantom. And you can see there are like tubes running the blood inside inside these tubes. So which is pumped by the outside pump, right? There's a oxygenated blood and deoxygenated blood pumped through this phantom. And this is the multimodal imaging panel. So you see a B-mode ultrasound providing the structural information about the prostate and other regions. And you have a photoacoustic image imaging the ICG. We have an ICG tube also here. So you can see an ICG contrast as well as the hemoglobin contrast that is coming from the photoacoustic image co-registered with the ultrasound image. So because if you just show the photoacoustic image, people can't make out where the contrast is coming from. Just like PET CT, right? You need that CT reference to confirm where the origin of the PET signal coming from. The same thing here. We need that ultrasound anatomical information to confirm where the origin of the photoacoustic signals are. So, and then we also did regular Doppler ultrasound, which confirms that you can map the blood flow. You cannot see the ICG inside the Doppler ultrasound because the contrast is only coming from the moving particles inside the blood. So, and we also did elastography information. We put a little target. We made the tumor region stiffer here by developing a biomaterial here. So, and you can see that we can map the elasticity. So this is what we envision, right? So that's how we are pushing the technology here. We are making both our instrument stronger, multi-parametric, that it can, one single device in one single setting, right, can provide all this kind of multi-parametric information. At the same time, from the other side, we are also developing contrast agents that can help us provide multimodal contrast. And in particular, we are hoping that when we conjugate them with certain, functionalize them with certain peptides, so they can help us target or get the more specificity from the prostate cancer biomarkers. So that is with this. I'm going to, I don't know how I'm doing with my time, but I'll try to wrap up quickly. So we are also working on
Yeah.
brain cancer applications. So my student Hao Young Chen and Shubham Merk, they developed this new technology. So where we can combine uh, a functional ultrasound or a power Doppler ultrasound, which provides beautiful microvascular information, right? And with the photoacoustic information to provide multi-parametric imaging. For example, here, this is a conventional ultrasound probe. We integrated the light guides next to it and to enable this kind of multi-parametric information. In the middle is what is called super resolution ultrasound imaging, which is microbubble enhanced super resolution ultrasound imaging, which helps us map even the tiny microvasculature inside the brain. So this is all in vivo imaging, and so we want to use multi-parametric. Here, the multi-parametric is coming from, so the power Doppler ultrasound provides the blood volume changes. The super resolution ultrasound can be used to quantify the microvascular density, because you can see all those fine vasculature. You can quantify the microvascular density. So tumors are supposed to have this angiogenesis, and from there if you can quantify the microvascular density, it acts as one of the intrinsic biomarkers to detect the tumor. And we can also provide the photoacoustic based oxygen saturation, right? So in addition, these animals can be subjected to hypercapnia, which is a short CO2 stimulation, and which can help us assess the vascular reactivity. And we want to see if this can be used to, for example, here you can see that by monitoring the power Doppler ultrasound changes as well as the photoacoustic based oxygen saturation changes. So when the CO2 is off versus CO2 on, whenever the CO2 is on, the vessels dilate, and there's a more blood flow overall globally. So we want to see if the blood volume is changing and whether the oxygen saturation is changing, how the oxygen saturation and blood volume is changing. In particular, whether the tumor regions have any differential response compared to the rest of the brain in terms of the blood volume or in terms of the oxygen saturation, right? So with that hypothesis, we injected the brain cancer cells deep inside the mouse brain, right? And these are like orthotopic, brain tumor models. And this is a very new information. So this is not published, but we are hoping that these novel results will help us go into high impact journal. So what you see here is a power Doppler ultrasound, and you already start to see that the contrast from this particular region of the brain is somewhat abnormal, right? And same thing, the ultrasound microbubble based super resolution ultrasound also confirms that these vessels are more tortuous, right? You can also quantify the vessel tortuosity, not the microvascular density alone, but you can also even quantify how much the tortuous are vessels. So you can clearly see that vessels are more tortuous or more abnormal here, even in the super resolution ultrasound. And photoacoustic imaging also shows that these tumors are somewhat hypoxic. And when we induce the hypercapnia, right, the vessel reactivity also shows that these tumors are having a different types of vessel reactivity, as you can see from this video, right? So when the animal is subjected to a short CO2 stimulation in the tumor region, we see some hyperactivity there. So in some tumors, we see that, but when the same cell line is injected into a nude mouse, we see a different type of response. We are trying to understand where this is coming from. This is actually an opportunity to study the tumor heterogeneity at a very high spatial and temporal resolution. So as I said, we are also, sorry to switch gears so fast. So we are also using this kind of portable photoacoustic and ultrasound technologies for imaging vascular diseases. This is a grant that I have funded with IASC Bangalore. The goal of this project is to develop a portable instrument that can be used at point of care in resource poor settings to study the vascular diseases, right? So one of the fundamental strengths of photoacoustic imaging is that it is more sensitive to the blood flow, blood microvasculature compared to the regular ultrasound. And because you are imaging the vasculature here based on light absorption of hemoglobin, as opposed to in ultrasound, it is based on blood flow mapping, right? So we want to combine these two contrasts. So blood flow, ultrasound-based blood flow mapping, as well as photoacoustic-based oxygen saturation imaging, and as well as another important information photoacoustic can provide about the vascular disease is that the plaque information. So the vascular diseases are originated because there's a plaque buildup inside the blood vessel that obstruct the blood flow, and ultrasound is used to detect the blood flow. But the ultrasound is also kind of insensitive to the blood flow, especially when the blood vessel is deeper, and also that assessment is operator dependent. And it doesn't provide direct plaque information. It only provides blood flow information. Therefore, by combining photoacoustic imaging to the ultrasound, we are hoping that we can provide a better assessment of the plaque formation, because the plaques can absorb light at certain wavelength and can be imaged. So overall, this is a project in progress. So we have a portable instrument such as what you can see here. It's the commercial LED-based photoacoustic instrument. Right now, we have developed even a better instrument that actually is much better than this commercial LED-based system. So we initially studied how well this can image the vasculature in collaboration with this Cyberdyne company, which sells this photoacoustic system. So as you can see that compared to a laser-based systems, LED-based systems can also provide equal quality. So laser, as I showed you before, in the prostate cancer system, that footprint is really bulky. So you have a big laser, but if you want to use something for resource poor settings, you need to use a portable laser source, light source. So therefore here we use the LEDs. So yeah, this is a project that is also ongoing, and the future directions. So my lab works at the interface of all these three circles that you see. So we develop theory and simulations of how light propagates inside the tissue, how the ultrasound propagates inside the tissue. So we develop this theory as well as imaging simulation platforms for all these three types of imaging modalities. And we also work on novel hardware development. We develop a new type of transistors, which are light transparent ultrasound transistors that can be deployed more effectively for multimodal imaging. And we also develop model-based AI systems, right? So for example, I just want to leave with this last application. If you look at the conventional cancer treatment, right? So two types of technologies are widely used for detecting the cancer. One is ultrasound-based image guidance is used. For example, ultrasound endoscopes are inserted through different organs of the body, right? So to help diagnose the cancer. At the same time, optical endoscopes are also widely used. That could be bright field optical endoscopes, or it could be a fluorescence-based optical endoscopes. But these two endoscope technologies, right? These endoscopes are used at different time points on the same patient. So the patient has to undergo multiple endoscopy procedures that not only increases the cost, but also brings discomfort to the patient, right? So our rationale is, can we combine ultrasound endoscopy and optical endoscopy, in fact, even provide photoacoustic endoscopy all together in one single endoscopy system? For that, you need to develop a light transparent ultrasound transistor. So we are working towards that strategy. Here you can see this looks like a glass plate. So, but it's a ultrasound transistor array. Put it on the top of a Penn State logo. And we did a little phantom study to confirm that we are able to generate ultrasound, photoacoustic, Doppler ultrasound, as well as fluorescence imaging through this array. So this is something that we are highly focused on, and we are hoping that in the next generation endoscopes, would have all this integrated into together to gain more comprehensive information about the tumor. We also could implant this transistors on a mouse brain, and we can, which can, right? So which enables us to map the even at not just an endoscopy scale, we can also do improvements in the microscopy domain. So where when the mice are implanted with this transparent transducers, they help us image the awake mice using photoacoustic imaging, as well as you can also do 2-photon microscopy and intrinsic optical sensing. So we are in an emerging study, the same transistors which are implanted on a mouse brain, which look like a glass window, are able to stimulate the brain. So we can also induce ultrasound neuromodulation using the same transparent ultrasound transistors. As you can see here, when we do the ultrasound stimulation during this period, so you can see we are able to suppress both the hemodynamic activity as well as the neuronal activity in the mice brain. So this is something we are interested, and we want to see if ultrasound stimulation can help suppress the tumor activity. So people have shown that in electrical fields can suppress the tumor activity. So they help not grow the tumors, the brain tumors. We want to use this technology in future to see if this ultrasound stimulation can also be used to reduce the tumor activity so that the tumors don't grow that faster. So that can be confirmed by this novel multimodal microscopy imaging on top of the ultrasound stimulation. So yeah, with that, I thank everyone. So our lab, in summary, develops theory and simulations and imaging devices, and they are used for this multiple preclinical and clinical applications. I also thank all our funding sources, as well as the fantastic collaborators, as well as my lab students and collaborators. Yeah, thank you very much.
Great, that was a great speech, Dr. Raj. So here we have a couple of questions in the chat box. So let me take those questions for you. May I read them out?
Yeah. So should I exit or?
That's fine. I can read them out. It's necessary.
Oh, sure. I also see okay, let me see.
Yes.
Yeah, so Amol, thank you. So great and information. If MRI can detect micromolar concentration of biomarker, the strength of PET is that PET can detect nanomolar concentration. What concentration? So that's a very good question, Raghuveer. Yeah. So what our study confirms is that ICG can be detected at trace level. So at micromolar, definitely we are not in competition with PET imaging in terms of molecular sensitivity. So, right. So, but there is a need for image guided, because transrectal ultrasound is the gold standard. And to guide the needle biopsy. So therefore, we want to attach the photoacoustic imaging to that, and then do this molecular imaging. Definitely, it's not as sensitive as PET, but we believe that we are in the micromolar range if the PET is in the right nanomolar range. And MRI, I think, is in the millimolar range.
Raj, can I add something or ask something in parallel to that?
Sure.
So yes, transrectal ultrasound is the gold standard currently in the US. But if you look at the rest of the world, especially in certain parts of India, they have actually moved from transrectal ultrasound to PSMA PET CT guided biopsies of the prostate, and they're actually using a transgluteal approach for that. And I was curious if there are any thoughts to combine the molecular strength of PET with the focused acoustic probes and somehow even get the best of both worlds.
Yeah, so you're right. Actually, the paper, I did not show that result, but in my paper, we did combine that. But that is the hopefully I do that at a larger scale. That is the vision of Sam Gambhir. So,
Mhm.
we did actually in this study, although it's not shown here, the PET imaging result. So we did combine the bombacin-based PET imaging with MRI-based T2 contrast or T1 contrast-based MRI imaging, as well as photoacoustic imaging.
Awesome.
So,
Awesome.
so in this study, for me, it's a need because I still see photoacoustic as a baby, which needs a helping hand of PET imaging and MRI imaging initially to confirm what I see in the photoacoustic is in fact the cancer region, right? The biomarkers that I'm seeing. So
Mhm.
therefore, I'm taking the guidance of PET imaging as well as MRI imaging, and then correlating that contrast with the photoacoustic contrast.
Awesome.
Yeah, so I did not show that here, but there is a bombacin result we have shown in the paper, in the Science Translation Medicine paper.
Perfect.
Excellent and informative tool and very knowledgeable. Okay, what is the accuracy and efficiency of developed device? Can it be used for early detection of cancer? Yeah, Bhavna, that's the hope, right? So yeah, that's the study. I mean, we have got fortunately this grant now, and you will be part of that. So because you are going to help develop the dual modality contrast agents and test them for in the preclinical models. So that's what how accurate this technology can be, or how sensitive this technology can be for. So yeah, that's what hopefully in the next 5 years we have answer to that. Okay, photoacoustic is cheaper and faster, but no, it's not less resolution. It's in fact higher resolution than PET. PET provides a typical resolution of 2 millimeters. The photoacoustic is sub-millimeter, like 0.5 millimeters typically. So we have a better resolution than PET. But the molecular sensitivity is not better than PET.
Um, Raj, so there's also the issue of depth for photoacoustic, right? The resolution is depth dependent. You can
Yeah, you're right. That's why I said
that's a trade-off, right?
yeah. So the 0.5 millimeters depth I said is about covering the entire prostate, which is about 5 centimeters depth.
Okay. So is that a good resolution right now for the depth? Like, for that depth, you'll get 0.5 millimeter resolution. Because
I
generally we when you're imaging cancer, you're not imaging just one organ, because I mean, so the focus seems to be on detecting a lesion, whether it is cancerous or not. But that's just the beginning of the journey for management of cancer, and then you do need additional things to stage it accurately. So I mean, this would help if we as a human race are so evolved that we're picking up lesions in very early stage. But eventually, you do need to pick up disease that has spread. And I think there lies the issue with acoustic imaging, because the depth, especially in the U.S. population, which tends to be, their physical characteristics tend to be, body habitus is challenging for acoustic imaging. So I
yeah.
was curious, what's the trade-off, and what developments do you foresee to overcome those trade-offs?
Yeah, so that's a very important question for acoustic imaging field. Just like optical imaging, right? For acoustic imaging suffers from the same limitations of optical imaging, but not to that extent. So our imaging resolution and quantitative accuracy, whatever parameter for acoustic can quantify, oxygen saturation or ICG concept, is everything is depth dependent. So, right, so that's why one of the things we provide in our publications is how this resolution and molecular specificity is changing as a function of depth. Especially this is challenging for applications such as prostate, where you have limited space for how you package the light and sound. For example, in the breast imaging, where you have access to the breast from all regions, they have a better resolution and better quantitative accuracy here. because you are able to capture all the photoacoustic waves generated from that, right? So you can provide a very good resolution. They can maintain an isotropic resolution of about, in this case, they have a 0.25 millimeter even at 5 centimeters depth, which is good enough, and that's what they showed. But for the prostate imaging, where your space is like 1 centimeter or 1 inch, sorry, so that becomes a little challenging to provide a uniform spatial resolution throughout the depth. So it's more or less 0.5 millimeters. So I'm more worried about not the resolution there, but to the molecular sensitivity, right?
Okay.
So how much of a ICG concentration I can detect. For example, when you inject the ICG intravenously into the humans, right? And how much of this ICG would accumulate inside the prostate cancer regions, and whether I am sensitive to that levels of ICG, trace levels of ICG. So our initial results shows that we can detect, but that study is only 10 patients. And so the new grant that we got gives us an opportunity to study, and we have an improved instruments right now. So for example, the study at Stanford, I had only 64 element array. Right now I'm using a commercial ultrasound transducer adapted with photoacoustic. So I cannot, I don't know, but I'm hoping that we can provide some useful information, because ultrasound is anyway used as a guidance. And needle biopsy, you cannot rule out, although the PET imaging, MRI imaging, all these beautiful technologies are emerging to detect the prostate cancer, and we need them pre-procedural and post-procedural compared to the trans. So you need that ultrasound guidance, because you need that biopsy to be sent to the pathology to confirm the cancer, right? And the best way that's why they do MR guided biopsy. Now the gold standard in US for sure is all hospitals are using pre-procedural MRI. That means a patient goes to the MRI clinic, and that MRI information is loaded on the transrectal ultrasound, and the biopsy is done in the guidance of the MRI. But we still see that there are many papers shows that this information is not still not sensitive and still not specific. So therefore, we want to further add photoacoustic, which is truly integrated to the ultrasound compared to the MRI. So there are some co-registration issues with MRI and ultrasound. So therefore, we hope that photoacoustic can still help the prostate cancer biopsy.
Awesome. I think there's another comment or question.
Yeah.
In the chat.
In the chat box, we have another question.
Has photoacoustic imaging used for colorectal and stomach cancers? So not stomach cancers. There is a technology being pushed for colorectal cancer. To combine that with, I see like multiple types of endoscopy imaging systems emerging, especially multimodal endoscopy systems combining conventional ultrasound and optical imaging technologies. They have done transvaginal ultrasound. There is a Dr. Quinzu group at Washington University, St. Louis. They are also well funded. They are studying the ovarian cancer. I see melanoma cancer, many people using. Recently, Crohn's disease activity. There are works for using photoacoustic for inflammatory bowel diseases. But I have not seen somebody using it for colorectal.
For optical stimulation, what is the nanogram angstrom units you use, or which range of light photons are used?
So okay, yeah, that's sorry, I did not mention that clearly. We prefer in the near infrared region something like 750 nanometers to 950 nanometers.
If you use ultraviolet in a much lower angstrom units, will you think that your detection sensitivity will increase?
Okay, so the longer wavelengths will help penetrate deeper. that's why we avoid, that's one reason we avoid ultraviolet. The second reason is ultraviolet itself can cause cancer because it's right, it is absorbed by the DNA and RNA, right? So therefore, people try to avoid using ultraviolet for imaging unless there is some endoscopic uses. Some people use ultraviolet based endoscopy because they are not in contact with skin. So for a reason, because there are NADH and ADH are the molecules which can provide metabolic information about the tumor. So they provide autofluorescence. These molecules NADH and ADH can provide autofluorescence when you excite them with UV. So this is one clinical application where I see UV-based imaging of tumor activity using endoscopic, but otherwise people try to avoid ultraviolet rays. But yeah, that is one reason, and then they won't help penetrate deeper. And blood absorption is also not that great in that region.
Yeah, even X-rays and gamma rays can cause cancer, but that has not stopped us using it for diagnostic purposes.
Yeah, the dose, the dose becomes, but that, as I said, that's not the main reason. So the depth penetration, if you use a shorter wavelengths, you cannot penetrate deeper. That's why even
that's a good point. But for a superficial lesion, probably I'm just thinking aloud whether it can increase the molar at what level concentration you can detect the signals.
yeah.
If you use a lower range wavelengths.
Yeah, people, so there is like there's another emerging applications of photoacoustic imaging is label-free histopathology. Normally, when you do the pathology, right? So you biopsy the sample, they send this to pathology. The tissue is stained to confirm different biomarkers, right? So what people in photoacoustic are doing is called label-free pathology. So you put the biopsy specimen and you do a high resolution photoacoustic microscopy on that to image the internal structures of the specimen. So in that, they use UV light. When they do the pathology on samples, they use the UV light. So there's but not in vivo. I have not seen anybody using for ultraviolet for in vivo, but because of the reason, like, you cannot penetrate deeper.
Yeah.
Awesome.
Thank you.
I do have maybe like a basic question about the photoacoustic thing phenomena basically. So as it's obvious in lightning and thunder, there's a lag between the photo part and the acoustic part. And I assume that's the case even at a microscopic level in tissues, that there's going to be, it may not be in seconds, but it will probably still be in microseconds or nanoseconds. And since we're looking at micrometer and nanometer resolution, that lag may still affect the resolution of the system and may pinpoint you in a different spot. Does that affect your resolution at all, and how do you get around it?
I like the question. This is first time somebody relating the resolution to the lag actually. So I don't know your background, but that's like typical like engineering level of thinking. But yeah, so it doesn't affect because as I said, so it is a nanoscale, the time lag between the light incidence as well as the acoustic, generation and propagation is at nanoscales, because we are not looking at such a dynamic events. So let's say if you are looking at such a dynamic event, that are happening at a micron scale, right, microsecond scale, then you may be affected. But we are not looking at, for example, even the brain activity is happening at millisecond scales, and the tumor activity, right? So we are mostly in the tumor, when we are detecting the tumor, we are mostly detecting the static activity, not like, that changes at microsecond scales. So for example, I injected ICG. So I know that it takes like about 30 seconds to get accumulated, right? Even if I image 1 second, if I take one photoacoustic image per second, I'm still fine with doing my molecular imaging with photoacoustics.
Okay.
Even when I'm monitoring hemodynamic activity in the brain, which is most likely happening at millisecond scales, right? So I'm fine.
Yeah. Yeah, I do happen to be a biomedical engineer as well in my past life. I don't practice that much, but
Yeah,
I'm
that's
not
like,
completely nuclear medicine, but I did do and this was a fantastic talk. It took me back to my own grad school days at UVA, where we were working on microbubbles, and we were using targeted microbubbles at that time.
oh yeah.
There was no photoacoustic, but we were trying to coat the surface of the microbubbles with targeted ligands that would bind specific receptors, or P-selectin, L-selectin for inflammation, and so on and so forth. So that it was interesting for me to see your data on nanobubbles and microbubbles. When you hit those bubbles with an ultrasound pulse, they burst, right? So, how do you know that the signal you're getting? Because you said that you incorporate the ICG within the bubble and not so it's not binding to anything till it's burst and released. So how do you know? So you're getting two signals, one from the microbubble or nanobubble contrast for the ultrasound, and one from the photo part of the ICG itself.
Yeah.
And so how do you know that the signal from the ICG is not from it just being released in the environment because of the bursting of the bubble, and it's specific targeted?
Yeah, so one thing, we are not bursting the bubbles. So people do that for blood-brain barrier opening and all. Um, we send very low intensity ultrasound pulses, including, so we are using also, why I say with very good confirmation is that because even in the this brain imaging results that I showed, this is based on tracking the microbubble for a long period of time, for two
Oh,
or
okay.
3 months. So this is how we generate such a, this is nothing for acoustics here. It's microbubble enhanced super resolution ultrasound imaging, right? Where we inject microbubbles intravenously and we track its movement through the brain using localization principles. And so as from this result and also previous results, so we don't burst the bubbles. So we use low intensity ultrasound. People do that for a reason, for therapeutic purposes, that they want to release something from the bubbles. Yeah. But here we are not. So they are fine.
No, even for diagnostic purposes, so the FDA approved microbubbles, the indication is to burst it to get a signal that acts as a contrast for ultrasound. So
Oh,
this
that is
is
a, oh yeah, I understand what you're saying.
yeah,
That's a
yeah,
non-linear ultrasound. We are
yeah,
not
yeah.
using that's when you want to work in the non-linear. So people are using, you're right. People are also using microbubbles in a non-linear regime where they burst the bubbles to further enhance the non-linear contrast. But with this emerging ultra-fast ultrasound that provides this kind of super resolution imaging, right? We don't have to do that, hopefully.
So having the ICG within the bubble, does the shell of the bubble affect the photoacoustic effect of the ICG itself?
I don't think so. ICG is inside so far, right? So that's something microscopical. This is actually, we are working with Dr. Agatha Exner from CASE. So her group is also working other groups to look at things microscopically, such as your questions, how the ICG, or how do we, how this dual modality bubbles, whether putting ICG inside the bubble would affect the ultrasound contrast. They are also looking at that. But from what we see, we do, I mean, obviously, if you have a bare ICG, you have a stronger signal, but ICG sticks to the vasculature. That is very peculiar property of the ICG. So it is unspecifically binds to the vasculature. So therefore, the amount of ICG that you can deliver to the tumor is very, the possibility is very less. So therefore, our idea is that by putting the ICG inside the bubble, so there is a better, that way we can deliver the ICG to the tumor more effectively. But whether it is affecting or it is getting affected by that, maybe perhaps, but still we generate a lot of contrast. In fact, actually, let me also tell you that. So in the nanobubbles, we see a better photoacoustic contrast. We see that fluorescence is getting quenched. So in this region, if you look at this result, in the nanobubbles, ICG is more packed within the small volume compared to the microbubbles. And you see that photoacoustic contrast is very high in the nanobubbles compared to the microbubbles. So there are different hypotheses for this. One of that is the fluorescence of these molecules are getting quenched. So that is resulting in more heat emission, which is translating to more photoacoustic signal generation.
Awesome. One last question. I was pretty intrigued by your plaque thing in the extremities in a resource poor situation. I think US is also headed for a research poor situation given the way things are fiscally in the healthcare environment. But that apart, in addition to imaging the plaque, can it be used to image a clot in a vein?
Oh yeah. Oh yeah. So we do see many people are working with photoacoustic, the thrombosis, right? So
Mhm.
the clot has, because photoacoustic is intrinsically sensitive to the hemoglobin molecules. So both oxy and deoxyhemoglobin, and then ultrasound is very sensitive to the flow. Even photoacoustic is sensitive to the flow. You can actually detect the clot information more specifically.
And then can you translate this to other body regions like the carotids in the neck or the coronaries on the heart? The heart may be more dynamic and tricky, but the carotids may be another easy target for the plaque.
Yes, there are. I used to show some applications with photoacoustic. There are people working on carotid artery imaging with photoacoustic. They have been translated to clinic. I'm not focusing on that, but there are few groups working on that. And for heart, it is very tricky unless we do intracardiac ultrasound, uh, and you do put photoacoustic in that intracardiac ultrasound. It becomes tricky otherwise from imaging from the um, non-invasively imaging is tricky. But for carotid arteries, yes, there are people working on that.
Awesome talk. And then one of the last things would be to look at urinary bladder cancers, because early detection, especially muscle invasiveness, is still challenging non-invasively as of now, and that may be another application one can focus on with the acoustic probes actually. So
Yeah, so can you please say it again? Sorry.
I said urinary bladder cancer is another
Oh, yeah.
potential application, because right now, early bladder cancer, we don't have good imaging tools. Even with MR, we can't detect with certainty whether it is muscle invasive or coming outside and so on. I mean, coming outside, yes, we can, but muscle invasiveness is not that well detected. And the bladder tends to be a dynamic organ based on the bladder wall thickness changes based on the fullness of the bladder and so on and so forth. So we are lacking good tools for the T stage localization of the bladder tumors. And I think since this is probe based and you're already in that area with transrectal and cystoscopy approach is also possible, although the apertures are smaller there, so you might have to develop miniature probes. But that's another area of potential where it can have a significant impact in the management of cancer clinically. That's
Yeah,
another.
I do agree. People are working on transurethral. I don't know if that helps in.
Definitely, it will help.
Yeah, people are working towards. I mean, because they have shown that they can develop a 3 millimeter endoscope. The transparent transducer, which is a flagship innovation we are leading a lot of innovation in that. A transparent ultrasound transducer becomes a critical component when you want to miniaturize things into a small form factor. Because otherwise, if the light and sound share the same space, right? That's what is enabled by our transparent transducer. Then you can develop more compact system. Even we are actually trying in one application we are exploring especially for neurosurgery. We want to integrate this ultrasound photoacoustic catheter inside the surgical needle.
Sounds good.
So that's like to help neurosurgeons detect the blood vessels or to avoid the damage to the blood vessels during neurosurgery.
Yeah. Excellent talk. I think in a sense of time, it's 11:30, so past half hour, but good. I was very intrigued by all the potential applications. This is something that we don't get much to hear on, so I'm very glad that you agreed to talk to us on our group. This will be recorded and put up on our YouTube channel as well. And thank you, Vani, for your kind words. Yes, it is indeed very heartening to continue the legacy of Sam Gambhir. Totally agree with you.
Yeah, I'm very fortunate to, I want to like take this opportunity also to thank everyone for giving me Dr. Josna Garu. I know Dr. Josna for a long time, and because of Sam again. And Sam has been my great mentor, as well as also my PhD advisor, Li Hong Wong. So you are nothing but, your mentors, vision that you carry. So I'm very happy to see, I'm Sam's lab is to be called Multimodal Imaging Lab, right? So I'm doing that in a little different way. Um, but I hope that there will be some patients benefited with our technology in near future. So
Awesome.
thank you for everyone for giving me this opportunity. I hope to join in some of the upcoming seminars. Thank you again.
Thank you.
Okay. Yeah, thank you. Thank you, Dr. Raj. That was an exceptional presentation. Your insights and expertise made us, gave us a significant impact, and we're all grateful for your valuable contribution. So thanks again for sharing your knowledge and making this event a big success.
Oh yeah, thank you very much.
Mm.